Welcome. Today we're going on a pretty incredible journey. And no, not to some distant galaxy, but deep inside ourselves. We're about to explore the bustling, invisible cities that make up our own bodies, and the absolutely revolutionary technology that's finally giving us the map.
I mean, just think about it for a second. Our bodies are these unbelievably complex ecosystems, right? Trillions of cells acting like individual citizens, all forming neighborhoods, communicating with each other constantly. For decades, if scientists wanted to understand what was going on inside a piece of tissue, say a tumor, they had to use some pretty blunt instruments.
You see, the old way was basically a blender approach. And yeah, I mean that literally. You take a piece of tissue, grind it up, and then analyze the molecular soup that came out. Sure, you could identify all the components, the proteins, the RNA, but you lost all the context. It's kind of like trying to understand a city by analyzing a smoothie made from it. You know there were buildings and people in there, but you have no idea where the financial district was or which neighborhoods were hotspots for activity. You've completely, totally lost the map. But what if you could have the atlas instead?
Well, that atlas is no longer science fiction. There's a revolutionary field of technology that's giving it to us, revealing that biological city in all its amazing complexity. This is the big reveal. This is spatial omics, a set of tools that are fundamentally changing how we see life itself.
So what is it really? To put it simply, spatial omics lets scientists measure all the key molecules of life. We're talking proteins, RNA, the works. But here's the absolute game changer. It keeps their spatial information. It's like giving every single molecule its own GPS coordinate inside the tissue. We don't have to choose anymore between knowing what is in our cells and knowing where it is. We finally get to see both at the exact same time.
And trust me, this isn't some niche, futuristic idea. The scientific community has recognized its power in a huge way. The super prestigious journal Nature Methods has not once but twice crowned this technology Method of the Year. First, back in 2020 for mapping RNA, the instruction manuals of our cells, and then again, just this year in 2024 for mapping proteins, the actual machinery that does all the work. So yeah, this field isn't just coming, it's already here.
Okay, so how in the world does this molecular GPS actually work? The tech is incredibly sophisticated for sure, but we can really get our heads around the main ideas by looking at two major strategies, two different paths scientists can take to build these incredible maps.
So these two main approaches come with a classic trade-off, right? On one hand, you've got sequencing-based methods. Think of this like casting a really wide net. The goal here is to measure pretty much everything that's there, capturing thousands of different genes at once. The trade-off? The resolution is a little lower. One data point might cover a few cells. Then, on the other hand, you have imaging-based methods. This is more like using a high-powered microscope. The whole point is to see exactly where things are, with stunning single-cell or even subcellular resolution. The catch is you're targeting a smaller, more specific list of molecules.
All right, so let's zoom in on one of these paths. We're going to focus on imaging-based spatial proteomics. This is the tech that maps the proteins, the real workhorses of our cells, and it is already making a massive impact in areas like cancer research.
A key technology that's really driving this whole revolution is called Imaging Mass Cytometry, or IMC for short. It's a really brilliant method that combines two powerful technologies to do what used to be impossible. See dozens of different proteins in a single tissue sample all at the same time.
So here's how it works in a nutshell. First, you tag your proteins. Scientists use antibodies, which are kind of like molecular homing missiles, that are designed to find one specific protein. But here's the trick. Instead of a normal tag, each antibody is attached to a unique heavy metal ion. Step 2, you scan the tissue with a super precise laser. This laser zaps tiny spots on the tissue, vaporizing them, and sending those metal tags flying. And finally, you build the map. A machine called a mass spectrometer figures out which metals came from which spot on the tissue. And since each metal corresponds to a specific protein, poof, you can reconstruct an incredibly detailed protein map.
And get this, it's not some slow, laborious process anymore. Modern systems like the Hyperion XTI have totally supercharged this whole workflow. They can run this analysis with incredible speed and automation, with some machines able to just load up and process 40 slides all on their own.
But what does that speed actually mean in the real world? Well, as Dr. Hartland Jackson puts it, faster means more samples, more data. And that's not just some small technical improvement. It's a huge leap in the scale of research.
It lets scientists analyze way more patients and get to those critical answers much, much faster.
Okay, so we have this amazing technology. We know it's fast, but what's the real-world impact? What does it do? Well, let's look at how it's being used to map the battlefield of one of our most complex diseases, cancer, specifically melanoma.
So, immunotherapy has been a miracle for some cancer patients, but honestly, a frustrating dead end for others. In melanoma, for example, over half of the patients who get treated with these powerful drugs that are designed to unleash the body's own immune system, they just don't respond, or their cancer comes back. The billion-dollar question has always been why, and spatial proteomics is finally giving us the clues we need.
And what researchers are finding is just fascinating. It turns out it's not just about which cells are in the tumor, but how they're all arranged. They can actually identify what they call cellular neighborhoods. For example, they found immune-hot neighborhoods, where the key cancer-killing T cells are surrounded by their support crew, and the presence of that neighborhood is a powerful predictor that a patient is going to respond well to therapy and have a much better chance of survival.
This just brilliantly illustrates how critical location is. Other studies have drilled down even further. They found that, yeah, having lots of cancer-fighting T cells is great, but what's even more crucial is how close they are to the actual melanoma cells. The closer the good guys get to the bad guys, the better the therapy works. It's so clear, in the battle against cancer, location is everything.
So what's the big takeaway here? This isn't just cool biology. This is a potential roadmap for personalized medicine. By mapping a patient's tumor, doctors could one day predict how they'll respond to a specific treatment, guiding them to the best option right from the start.
And as powerful as all this is today, we are really just scratching the surface. It's mind-boggling. So where is this incredible mapping technology headed next? What does the future of the map even look like?
Well, the horizon is incredibly exciting. Researchers are already working on moving from flat 2D slices to full 3D atlases of tissues. They're developing ways to layer multiple types of information, not just proteins, but RNA, DNA, even metabolites, all onto the same map. And for many, the ultimate goal is to contribute to global efforts like the Human Cell Atlas, this hugely ambitious project to create a complete reference map of every single cell type in the entire human body.
You know, for centuries, biology has been a bit like exploring a new continent without a map. We could see parts of it, sure, but we didn't really understand how it all fit together. Now, for the very first time, we have the tools to draw that map in exquisite, unbelievable detail, which really just leaves us with one final thrilling question. Now that we have the map, what new worlds will we discover within ourselves?